Well, good afternoon. Um, I really feel like I'm among friends here. I think I know so many of you. And so I was just thinking, let's just forget the talk. Let's go for, a glass of wine. But since we need to go and have the talk, we shall. It is particularly of importance to me that I'm talking here at the Simons Foundation. I would say that about 70% that I have to present to you wouldn't have been possible if it were not for a few conversations that we have a few years ago, where this foundation and the people who founded it actually believed that the kinds of things that we were attempting to do at the time were possible. And so I think I'd love to say to a couple of them, one of them is here, we delivered on half of the promise. At least half. The other half is just about to be submitted, so just wait. I'm gonna be talking about redefining autism, or an attempt to redefine autism. So I have to clarify one thing first, which is this notion that I'm a clinician first, always was, always will be. So the definition of autism as we have in the Diagnostic and Statistical Manual in the National Classification of Diseases is extraordinarily important. It's extraordinarily important because I live in a world, a healthcare world, an educational world, a legal world, in which those diagnoses matter a great deal. So this is your passport for eligibility for services. This is your passport for a whole range of things. The issue is not that. The issue is that we need somehow to conquer the enormous divide that I believe, and I say this with trepidation in front of Jerry Fishback, but with trepidation that we are a little bit at an impasse. We have had tremendous, tremendous advances in molecular genetics, but we're still trying to make a connection between those advances and autism. And the way that we experience children, their strengths, their needs. And I think that this is a way that we might be able, in our center, to maybe contribute something, is there's something in between genes and symptoms. And I think that that's what we would love to be able to make a contribution to. So it is in this light that we're talking about attempting to redefine autism. We see that the symptoms are kind of the endpoints, the outcomes of processes that really begin much, much before people like me can reliably diagnose the condition, much before that diagnosis is actually stable. So this process is something that has begun much before the age of 24 and 36 months. And I believe that if we want to really advance systems biology in which we have behavior and we have genetics and we have brain, we need to bring it all together. We need to conquer that impasse. We need to go earlier in development, more proximal to the time in which those interactions, those iterative connections are actually happening. So it is in this sense that I'm talking about redefining autism. But I have to, first of all, to thank all the children and families that have contributed to the research. Thank you, John, for mentioning my age indirectly. We've been in this field for a little while, and we have many people to thank. The other person that I have to thank is Warren Jones, somebody who's worked with me since 1999, and a lot of the work that I'm going to present is really a partnership, probably the most beautiful intellectual partnership that I've had with anybody in my life. And then all of those foundations that have been very generous in their support of our work. So, a second here. Slightly better. So first, I'd like to boast a little bit about our center. So this is the Marcus Autism Center, and is a 90,000 square foot building and an 18.9 acre lot about half a mile away from the CDC, about a mile away from the medical center at Emory. This is on a Saturday morning. There are 262 parking spaces there, and they tend to be filled very quickly. So let me tell you what it is. The Marcus Autism Center is the largest center of clinical care for children of autism and their families in the country by a factor of several. Over 5,600 unique children seen there over the calendar year 2013. we are not only a diagnostic and assessment center, we're also a treatment center with a whole range of treatment programs that really range from skill acquisition through severe behavior units, through feeding disorders, parent supports and parent training, you name it. And the Marcus is a center that was created about 21 years ago, but just recently, it was acquired by Children's Healthcare of Atlanta, which is one of the largest systems of pediatric healthcare in the country, with 1,700 pediatric physicians and a monopoly of pediatric healthcare in Metro Atlanta and the state of Georgia. So as John was mentioning, I was at Yale for 20 years. I directed the autism program there. I was a tenured professor. And it is truly a wonderful institution. But what was different about this place was, you reach middle age, and you'd like to know the extent to which your work has been relevant. Have you achieved tangible benefits to the children and families that you've dedicated your adult life to? And being part of a system of healthcare makes it possible for us to develop our new methodologies, our new technologies, to find new discoveries and see them being disseminated through the community, because we are part of a system of healthcare that is delivering those services directly to families. Now, Emory indicated their commitment to autism by creating a division of autism in the School of Medicine, which I chair. So we have the science on one side, and we have the system of healthcare in the other. All of a sudden, we saw that anything that we do within science can be brought to the community. So we could see that we could focus on translational research. As you know, all of us who have been working with NIH for many years, the big issue is what have we bought with the investments that we have made in research in autism. And so for me personally, the notion that from hypothesis creation all the way through the work that is done in our playrooms every day, we feel that connection between science and community, so that it would allow us to address autism as a public health challenge, which is what we wanted to do. And that was a little less possible within an environment that was more isolated from the community. Our goals of bringing together clinical research, advocacy, and training is reflected in every member of our center. There are 282. And one of the things that we have is the possibility of creating model programs of treatment within the center, but everything that happened in the center has a counterpart in the community. Because, needless to say, in this field, most of what we experience as autism, certainly in academic centers, is not necessarily what families experience all around the country. We talk about gold standards. The gold standards that were used, for example, through the Simon Simplex collection. Well, if you look at the number of assessments that happen in the community, less than 4 to 6% of those assessments in the community actually use standardized instruments for autism. Less than 5 or 6. So, repeat to you, good old ADI and ADAS is used in less than 6% of diagnostic assessments conducted in the community. So what happens with all the others? So this is what we came to sort of address. Well, what we have to address is the fact that, at Yale, which is a wonderful place, and I take responsibility, we had a 2-day assessment in which we deploy 5 or 6 clinicians. It cost $5,500. In many community centers out there, we have 25 to 30 children being seen between 8 and 4:30 in the afternoon. So what happens is that the reality of our children that come to those clinics is that the clinicians have time to do one thing: to make a note of the challenging behavior that they're showing and to dispense medication, because this is actually covered by the healthcare system. So this is a reality that we wanted to embrace, and that's the reason why not only myself but 18 of my colleagues moved from New Haven to Atlanta, is to address autism as a public health challenge. So But we wanted to capitalize on this huge center and make it also a center of science. And so the lines of science that we have delineated for ourselves in our strategic plan, so we research causes, we research treatment, but we also research community viable solutions, things that have to do with delivering services. And what I've learned as an advocate, being in DC probably more than in Atlanta these days, is finding out that we need to show not only evidence for efficacy of new treatments, we need to show also evidence for their cost effectiveness, otherwise they're not going to be disseminated. So we need things that are viable for the community. So that's the science that we want to practice. So we have 13 research cores, 9 are internal, really ranging from some of the things that I'm going to show you, all the way through developmental neuroimaging. But we also have collaboratives. We collaborate with Yerkes National Primary Research Center in Behavioral Neuroscience. We collaborate with Human Genetics. This is our genetics collaborative. We also, I just mentioned to Ruth a moment ago, we also created a bioethics collaborative because we feel we're dealing a lot of infants. We're looking at prodromal syndromes. We're dealing with risk communication. And now, if we have the ability to face a parent, and the child is between 9 and 12 months, and we are seeing the signs of autism before those symptoms are actually as clear as, as day, and it can be seen by a clinician. And you have a treatment that begins at the age of 12 months. And you are offering this to parents, and parents are not taking. They're not taking because they can't see the future. So this is called risk communication. This is now dealing with something that we might be able to identify in its prodromal quality. And so we need to really deeply research the issues involved, the bioethical questions involved. John was kind enough to mention that we are developmentalists. We are deep believers in what happens in the first years of life, particularly the first year, 2 years of life. This is the short period of time with the greatest postnatal brain growth and specialization that you're going to have in people's lives. So we better understand it. And in my sort of my time early on at Yale, I used to see a lot of babies. I saw hundreds and hundreds of babies in the first year of life. And I know that there are changes that happen month by month by month by month. So the notion that we could somehow cross-sectionally take one sample of a child's behavior at the age of 6 months and then wait until the child is 3 and try to gain some knowledge of that, this is not the way that the mind, that the brain, that development works. Thank you. You made possible for us to actually sample children's development 5 times before they're 6 months of age. And then every 3 months thereafter until 24 and 36 months. Because it's those kinds of intensive, longitudinal, prospective designs that allow us to actually come up with some findings that allowed us to see the unfolding of this condition before. And once we have something that is quantitative, something that is objectified, something that is mathematically allow us to say hello, to trace development over time, then we build an entire system around it, a strategic plan that goes from neurobiology to behavior neuroscience and neuroimaging and so forth. So this is the Autism Center of Excellence. We are one of the only 3 Autism Center of Excellence centers in the country. There's one in LA, one in Boston, and now one in Atlanta. And that, in a way, exemplifies what we're trying to do. Our first study, we're now developing new tools and new methodologies, new quantification methods to trace the development of what we call social visual engagement in the first 36 months of life with a risk-based cohort. These are siblings of children with autism who are, for whom there is a 20-fold increase of also having autism, and another 20-fold, so it's 40 overall, having a subthreshold form of autism. We also are tracing phonological and speech and language development monthly with those children using methods that allow us to sample the vocal environment in children's homes and then to develop algorithms that extract from those data set specific milestones in speech development in the first years of life. We have a randomized controlled trial that begins at the age of 12 months. And interestingly, the earlier the better. And because the kind of concepts that we're focusing on are evolutionarily highly conserved, and they are developmentally very early emerging, and because we know that the greatest influence on primate brain has been sociality, A lot of the things that we study actually are also very key for the adaptive survival of infant monkeys. And so with our colleagues at Yerkes, we are studying infant little monkeys from the time that they are born. We are actually studying them every week until they are 6 weeks, and then every other week until they become 6 months of age, using both behavioral eye tracking and neuroimaging techniques. More about that momentarily. We believe that this is a field in which there should be no competition. We believe that it's all about partnerships. The challenges are too huge for us to basically have institutions not collaborating. And so Atlanta has become, Metro Atlanta has become a place in which autism is a priority. I don't think that there is a department at Emory that is not collaborating with us or across Metro Atlanta. That we have many, because I have many friends, we have partnerships going on with all of those universities. Some of them are Simon investigators. And all of those funding partners that are helping us achieve our goals very, very, very quickly. So a little bit about autism. This is a crowd that is really very knowledgeable. But I would like to take a spin on this introduction of the condition. It's highly prevalent. Having said that, people need to remember that autism is more prevalent than all childhood cancers put together. So this is how prevalent it is, how much of a unique priority this needs to be for the community at large. And when people talk about the rise in prevalence, and there are all sorts of theories that are going on, people need to remember that one of the reasons the numbers have gone up so much is because we're finally getting things right. So if you look carefully into the CDC report, you're going to see that between that cohort, between 2002 and 2008, there was an increase of over 100% among Hispanic kids and 91% among African American kids. So this is not because there is an epidemic there, it's because we're finally reaching out those communities. So it's ascertainment, ascertainment, ascertainment. Another thing that I'd like people to think about is the societal cost of this condition, in excess of $130 billion in the US alone. But the number that really is closest to my heart is that one, that the lifetime cost of care for a very disabled child with autism is in excess of $2.4 million, and we have to thank David Mandel for having written that paper. So those are the kinds of things that should establish our priorities. Of course, it's a brain disorder. Of course, it's a genetic condition. It's the most strongly genetic of all complex neurodevelopmental disorders. Having said that, here we are. We have over 300 variants. People are, maybe 400, maybe 500. So one message that I'm bringing in out there is this notion that our genes, even our brains, are not as deterministic as to basically shape our lives from the moment that we're born, because we all carry mutations. And most of us, that I see in this room, we are not showing, big signs of disease. The notion is how that extraordinarily complex genetic liability translates into symptoms. That's the question that we would like to ask. How is this instantiated? Okay? Now, we know that early diagnosis and intervention is critical. I mean, the American Academy of Pediatrics has issued a recommendation that all children should be screened between the ages of 18 and 24 months, but there has been very low uptake. Two reasons, because now I have pediatricians all over me. One, they don't have the tools yet. They have questionnaires. Now imagine if my child, I'm where my child has cancer. I come to a doctor, and the doctor puts a questionnaire in my hands. That's not the level of medicine that we want to practice on a condition that can be potentially devastating. So that's what the pediatricians are telling us. And the other one is that, okay, if I identify, what are we going to do to those families? Because they still don't believe that we can do something for their families, and we can. The median age of diagnosis still hovers around 4 to 5. Only 20% of children who carry the diagnosis of autism into their school age, when they are now eligible for services and for IEPs, for individual educational plans, are identified before the age of 3. Only 20. So we're really missing this enormous opportunity that we have, that neuroplasticity affords us in the first few years of life. We have to do something. Besides, there are major community disparities, something that I was not close to when I was in New Haven, which is this notion that the age of diagnosis, it lags still in communities, in minorities, in rural populations, in poor families. And if early, better, it means that later you have more compromised outcomes. So we have whole sectors of our community that we are basically condemning to have worse outcomes. And then there is the reimbursement part of autism that this is not for this crowd, but I have to say that insurance has a long way to go for those families. The reason I bring this to you is because this is a concept that is clearly important if you're talking about autism as a public health challenge. The economics of autism is truly stupid. Because we wait to diagnose the children. By that time, we no longer treat autism. We're treating the burdens of autism. All of those things that our children, in fact, have learned over the years, including their severe behavior challenges. If you come and visit our center and you see our severe behavior unit, what you would find out is that there are 3 adults working with 1 individual in order to get that self-injury down. Because it makes it impossible for the child to be in the world, but it makes it impossible for families to leave. But it costs $70,000 to $100,000 a year. So, so this is what we're doing. We're waiting for our children to develop those highly challenging behaviors to treat them. Or we wait for them not to learn language, or we wait for them to develop intellectual disability. When I came into this field, I thought intellectual disability was entirely inevitable. I came into the field of autism by living in a residential unit for adults with autism. Those are individuals who were in long-stay hospitals for many years. I lived there for 3 years. All of them are profoundly disabled. All of them were profoundly intellectually disabled. And now that we've seen the babies come up, and what we're seeing is that that intellectual disability was not, if you're religious, it was not mandated by God. And I'm not sure the extent to which all of it comes from our genes either. But our children, they learn from others. And if that is blocked, they're not going to learn. Okay? And we have some data to that effect. So our goal, strategically, is to take that line of median age of diagnosis and bring it all the way so that we can actually put our finger at the time that this condition is forming itself, so that we won't keep on treating associated disabilities. We can begin to treat the actual core part of the disorder. And this is not a terrific slide, but here you go. This is our model. Typical children come into this world and they fall in love with those who are going to ensure their survival. It's not only human infants. If for those of you who know primates, that's the same. It's actually a whole range of species that require a caregiver to ensure the infant survival. And our kids tend to basically come with an attenuated sense of the other. And they end up falling in love with a lot of things in the world, but things nevertheless. And so, I'm quoting, because I'm in New York City, I'm quoting Joe Ledoux, who, the byline of his, one of his books, the subtitle of one of his books is The Way That the Brains Become Who We Are. So brains are also the repository of our experiences, and we should not forget that. We should not forget that. This is another not very good slide, but it gives you a sense of what we're talking about. You can go in that direction, or you can go in that direction. We all need expertise in both domains, needless to say. But we are horrifically biased towards people because we need people to survive from very early on. So, so what is kind of the model we're talking about? There is a genetic liability that this foundation probably has done more than most in advancing our understanding of that genetic liability. Those, however, are very unlikely to be connected directly to symptoms on the other end. And we believe that that liability has to do with disruptions of normative processes. If we want to understand that, we have to pay attention to the first years of life. So that's the notion. But what? So we go from here. We go with the notion that the platform for brain development is reciprocal social interaction, not only for human infants, but for other species as well. And just to remind you of what happens with the brain as we come into the world. And there's terrific organization that is experience dependent that happens in the first 18, 24 months of life. Okay, so. You're blocking the stuff you remember. You had to, you had to put those things in. So this is a 50-month-old little girl with autism who is entirely oblivious to me. And just this notion that we are in the presence of another, most of the time, we have an impact on the other. It's not happening here. So what is happening with her? 5 minutes later.
Haley, up. Haley, up.
5 minutes later, she finds out an M&M about 5 feet away from where we are. So she can see. She actually, at the time when we tested, she was not cognitively disabled, but the world is kind of dichotomized in her life. She doesn't see me 2 inches from her nose, but she does see other things. So I had to ask this girl, really, is she thinking about people? The reason being that brains don't have eyes, brains don't have ears, brains have experiences. And so what is she experiencing in others? We need to come up with a paradigm. So we came up with this paradigm, which is basically it's a classical paradigm to focus on one particular skill that emerges in babies as young as 4 days of age. And it is something that actually is very important for animals other than humans, including monkeys and dogs and cats, even in newly hatched chicks, because it's part of visual imprinting. So see if you get this. So this is called biological motion. This is the motion of living beings, particularly vertebrate animals. And what we have here is a point-light rendition of action. And so the idea was that we would have that animation going upright, and forward is basically a person trying to approach a baby. And there, on this side, you basically have the same animation but inverted, and we're playing it backwards. We didn't want an identical image here, a mirror image. And then we wanted to see where the babies would look like, and if they were able to match what they heard, which was the person's speech, to the upright. It meant that they were mapping this point-light space on a template of human action. And the notion is that the moment that you revert that, both from a mental but from a neuroimaging standpoint as well, the effect goes away. So what you see here is that little crosshair. That crosshair tells you exactly where that 2-year-old is looking at. And in this case, this typical 2-year-old was matching very easily and spent most of the time looking at the upright animation. So what about that little girl that I showed to you? She was random for the upright and the inverted figure, and which basically means that she was not differentiating one from the other. Something that is as foundational as that. And here are two little girls who are comparison girls, a 50-month-old and a 9-month-old, and they both showed a strong preference for the upright animation. When Warren and I were looking at those data at that point, we realized that there was one animation in which she did something entirely different. She was entirely upright in one of the 9, and this is the animation. And so we thought she got it right. So let's see what happened. What's special about this animation?
Patty cake, baker's man, bake me a cake as.
So you probably got it. There is a clap. Remember that the inverted animation is being played backwards. The speech, of course, only goes forward, and what is happening is that those lights are hitting, they are making a synchronous sound, but we created a asynchrony, dysynchrony happening in the inverted one. So our hypothesis became, oh my God, could it be that she's so oblivious to the social meaning of those animations, something so foundational for socialization, and yet she's so acutely sensitive to a physical contingency or the visual synchrony? And if so, then she would look where that clapping was happening. And that's indeed, this is her video. So this is exactly what she was doing.
Make me a cake for baby.
So we went on and we conducted a study. And a study that involved 3 groups of children. 2-year-olds with autism, 2-year-olds who are non-autistic but had developmental delays, and 2-year-olds who are typically developing. And this is what we found. Very much like in that case study, the children with autism were entirely random for those two kinds of animations, not so the typically developing and the developmentally delayed group. So, and for patty cake, which was that particular animation, we replicated what happened with that child And all of a sudden, the whole group of children with autism are showing that upright preference. And so we started sort of thinking, what was that girl trying to tell us? And here was the hypothesis that for the children with autism, behind the fact that overall they were random between those two animations, maybe their visual behavior was being guided by physical contingencies, audiovisual synchrony.
Ready?
Because when we talk, when we are talking to somebody else, we move in synchrony with our speech because we are one unit of communication. So we measured movement on the uprights, movements on the inverted, the audio changes, and what you have in those two here, in those peaks, are basically discrete events that mark audiovisual synchrony. This is patty cake animation, so you have a lot of audiovisual synchrony happening here and very little going on in the inverted one. And then we quantified that. We wanted to see how much more audiovisual synchrony there was on the upright relative to the inverted side of our animation for all of our animations. Once we did that, we could then look into the extent to which the visual behavior of these children had been driven by audiovisual synchrony. And what we found out, and look at the three at the top there, that 90% of the variants and their visual preferences were guided by audiovisual synchrony. Not so for the two other groups at all. Now this is a good journal, so they ask us take another cohort and conduct 2 additional experiments. And so this is what we did. And here is our regression line. If that hypothesis was right, then those preferences, oopsies. Something happened with that figure. You can go back to the paper. The 2 experiments, they fell over here in terms of the synchrony, and they fell right within the regression line, which basically told us that, indeed, the visual behavior of these children were being driven by a physical, not a social contingency. Now, why this is important, I bore you to tears with biological motion. But this is what is happening at the same time that we are doing experiments with our children. This is what they are doing. That little girl, when she was looking at a speaking face, like this caregiver, she was spending a lot of the time focusing on what?
Mommy, let's get up. Good.
She was focusing on that mouth. And here's the issue. The mouth is the place in the face where we have the most audiovisual synchrony because lip movements and speech sounds go vary. Okay? But remember, we are watching a person. And are you reducing that person to a composite of physical features? So when we looked at some of our eye tracking results that we got is that the children with autism spend much less time looking at people's eyes and much more time looking at people's mouths relative to the 2 other groups. But then we did the next step. And the next step is that we quantified the amount of audiovisual synchrony on those speaking faces. And when we did that, this is what we found. For the typically developing children who are on the other side, these are 2-year-olds, there is no correlation between their visual behavior and the amount of audiovisual synchrony at all. With the children, with the 2-year-olds with autism, what we found out is not only that we could predict that their looking at mouths was predicated on audiovisual synchrony happening in that face, but when they are looking at eyes as well. Now, that's an issue. That's an issue because the most important influence on a baby's brain development is the presence of their caregiver. And that caregiver is engaging that baby in this iterative process. So much of brain development depends on communication and so forth. And if our children, at least at those early ages, were reducing that important social cue to what is really a composite of a physical contingencies, then needless to say, their brains are developing differently because their experiences are being so fundamentally different. So we took that to signify that we had to go back in development. We wanted to create, these are growth charts. Growth charts of physical height and weight. We needed growth charts of social development, growth charts of social engagement. The reason being that maybe using that concept, we could push the possibility of identifying this condition from the time that those skills are online. And babies prefer to look at people who are looking at them rather than people that are not looking at them from the time that they're 4 days of age. By the time that they are 2.5 months, they fall in love with people's eyes, and that's what they look at and nothing else. So we knew that that was a critical thing, and disruptions of that normative processes becomes a marker of what eventually will become a symptom or a collection of symptoms we call autism. So that was the hypothesis. So we went on, and thanks to the Simons Foundation, we built labs that did not exist before. And this is my colleague Warren Jones. And with the technology that we had to put together, and this is a typically developing 5-month-old who is, happily looking at caregiver. At the same time that we're measuring the engagement of this 5-month-old with the world 60 times a second. Well, what did we learn? So we learned that there is a lot happening in those first months of life. We learned that if we were able to quantify something unfolding over time, that we might be able not only to identify the early disruptions, but also try and understand what was happening from a different standpoint, maybe from a biological standpoint. One of the things that we wanted to do is to have what people call an endophenotype, which is a process that it parallels the condition, precedes the condition, is not symptoms, but it's something that is quantitative enough upon which you can regress biological measures that somehow they should be commensurate to the unfolding of the condition. So that's what we're trying to do. And this grew from observations that we made. For example, that typically developing infant, one was looking at the caregiver, was looking there. The eyes are so important in socialization that the very morphology of the eyes were shaped in a way by selective pressures to be the way they are. Humans have the greatest amount of contrast between sclera and the color in the eye, and the lower you go in phylogenetically, species that don't require a caregiver, you don't have sclera at all. Now look, this is what a child with high risk for autism was looking at. It's a very different pattern, spending a lot of time on any movement happening in that scene as well as on mouth. So the idea was to create these growth charts, and we did. So this here is percent of fixation on regions of the screen, and here you have our data points going from 2 to 24 months. And so these are typically developing babies, and they come into the world, they prefer to look at eyes, they're not interested in mouth, body, and object, and this is what happens in the first 24 months of life. So let's take just eye fixation. This is what happens. It increases in the first 6 months of life, and then it becomes relatively stable. And this is what happened with the children who later were diagnosed with autism. They start way over there, and then you have free fall over the first 24 months of life. So we feel that this is the unfolding of the condition. At 24 months, we could ascertain the diagnosis, and it was stable at 36 months. Now, those are very different curves. And what you have at the top is the actual fixations. By the way, these are functional data analysis curves. So it's not your typical longitudinal growth curve analysis. And here what you have is the first derivative. If you look now at those curves, you start seeing that as we look at the first derivative, we are beginning to segregate those two populations. Now, the other fixation points are interesting to us because both are going down, as you see, for body, but you see that it was going down much more quickly, in a more steeply degree for the typical children. So much so that the moment that you look at the rate of change, you are segregating these two populations quite well. And what about object fixation? This, for us, was in a way, a metaphor for autism, because it's going down, but in the at 12 months, for the babies who were later diagnosed with autism, they're falling in love with objects. So by the time that they get to be 24 months, they develop expertise in objects. So we needed to see the extent to which there was clinical validity to this measure. And so in this functional principal component analysis, what you have the typical growth curve that you have for the whole group of children who develop autism, but some were more or less typical. And therefore, you can assign a principal component score for that. If we use those PC scores, we could, they were correlated at that level to what is a standardized level of autism, which is something coming from the ADAS. That correlation is 0.750. But then we wanted to become really, really quite ambitious here. Let's throw all the data out after 6 months. Let's just focus on the data in the first 6 months of life. So this is what we found. We found that if we used only the data of eye fixation and body fixation in the first 6 months of life, those curves were indeed very, very different. And then we wanted to take a look at the amount of overlap that existed between the two distributions. So remember, there you have the actual curves, and here is rate of change. And those ROC analyses tell you the amount of overlap between the two distributions, which is very, very little. And so we conducted, of course, leave one out cross-validation analysis for these groups, and we're getting major segregation between those two populations. And this is the result. Using those two at the same time, we could separate this group quite well. And then we took the next 6 children who participated in our protocols. When they were ascertained at 24 and 36 months, 4 of them were typically developing. 2 of them had autism, and we could classify those cases as well. Now, why I'm bringing this to you? I'm bringing this to you because maybe the time has come for us to sort of use methodologies that are going to allow us for high throughput screening of children. The reason we have those economic disparities there in our community is because we don't have ways, we don't have scientific ways of actually screening a high number of children so that we can identify this condition and we can offer these children treatments. So we needed to come up with a procedure that would be viable, that would be non-invasive, that would allow us high throughput, low cost deployment of universal screening. But also, by identifying them early, we would have maybe an opportunity to significantly attenuate this condition. So we set up in motion something that was made possible by our move to Atlanta, that will take this lab that was created thanks to the Simons Foundation and reduce it into something that could be deployed maybe in a pediatrician's office, like a prototype, a small footprint device that would allow us to collect data in primary care physicians' offices. The data sent to our center, where we can then deploy our data models and then analyze this data in order to be able to identify those children who are at risk. And we did that in a very aggressive manner. And so we moved from prototype to prototype. We built a building next to our center in 8 weeks. It's a shop where a lot of engineers and folks who came with me from Yale still. And now we are moving this. We now have a pre-submission to FDA, a large-scale clinical trial happening in 2015. We hope to have the results of this large-scale clinical trial. It's going to happen in 7 sites around the country by the beginning of 2016. Now, this is one thing that we liked. This was the result that I showed to you of eye fixation for the typically developing children in the first 6 months of life. But remember that 1 in 5 of those siblings will have autism. 1 in 5 will have something that is a subthreshold condition. It's called broader autism phenotype because we don't have really a definition. People are concerned about, and they are different from children who are typically developing in that sense. And we know very little about what is the outcome of those children. But for us, it was very important to take a good look at them because 18 of our high-risk siblings did not develop any diagnosis whatsoever, and 10 of them did. Okay? And it was interesting that those 2 groups, their curves were intermediary between the two extremes. But then we went further. Remember that what you have at the top is the amount of fixation, the second line is change, and the bottom line now, this is the second derivative. And the second derivative is the rate of change. If you're a biologist, one thing that you want to know is whether there are any step functions in, say, in the expression of a protein or something that signifies a change in the dynamic of the body, right? So this was the interesting thing for us, because if we take the 16 of the high-risk children, they had curves that were exactly like the normative ones. And those were children who did not develop a diagnosis. But if you take a look at on the right side, you see that the other 12, they started exactly the same way, and there seemed to have been a course correction. The course correction happened in terms of change around the age of 18 months, but the rate of change, the second derivative, the one that basically should indicate that something is changing in the person's body, actually began at the age of 9 months. So these children started exactly like the children who became autistic. But there was a course correction halfway, and they end up in a very different place diagnostically. So we were very excited about that. We're very excited about that because maybe then we could use this as an assay that truly goes parallel to the unfolding of the condition. And it would allow us, maybe with the regressors that we need in order to do things such as gene expression methylation studies, or anything that is going to allow us to measure the changes in neurodevelopment, in epigenetics, things that will now have a target. And the target is not a dichotomous one. The person has autism or doesn't have autism. Now we have a quantitative measure that unfolds during the prodromal period of this condition. And so if we take, as recently I had to write a kind of an essay in honor of Sir Gabriel Horn. Somebody, people might know Gabriel Horn as being the greatest maven of memory research using visual imprinting in the domestic chick. Well, that model is very much an inspiration for the kind of things that we want to do. Because a behavior that happens within the 24 hours of life of that domestic chick is associated with very significant and discrete neurodevelopmental changes, as well as gene expression changes. And that's what we want to achieve within the context of what we're trying to do. I'll talk about Williams syndrome in a moment. So we have a group of colleagues who are wonderful collaborators. We are part of the ACE network that focuses on the genetics of underrepresented populations. And we are gearing up our genetics portfolio right now. So one more thing that I wanted to say. When I sort of was coming to this field, we had the hypothesis that our children were born with this inability to relate to others. This is what Leo Kanner wrote in 1943. But if so, their percent of eye fixation should be flat and should really be low. But this is not what we got. This is what we got. So what's the meaning of that? In fact, this is statistically significant. So the children with autism actually started at a higher point. So we had to try and understand that. And in order to understand that, you need to understand the development of skills and reflexes in human beings. Not only this skill of being engaged to the eye of other, but most of other neonatal skills and reflect. They go through a period, the first weeks of life, they are reflexive. And they are reflexive, and they are really subcortically guided. All visual behavior occurring in the first weeks of life, given the maturational status of the visual system, is subcortically guided. And is experience expecting. In a way, there are specific biases that those babies come into this world and they want to pay attention to. And this period is followed a few weeks later by what is called an interactional but cortically guided visual behavior. This is something that doesn't happen only in humans. This is something that actually comes from Gabriel Horn's sort of visual imprinting model. That there are those two very specific phases that happen. Of course, the chicks' brains are very different, but the model also come from other model systems that are higher up in the phylogeny. But the notion is this, that at one point in time, visual fixation that babies bring into the world that is reflexive and subcortically guided becomes co-opted into a behavior that becomes interactional, reward-driven, and cortically guided. So we are basically hypothesizing that between the ages of 4 and 12 weeks, during that very narrow period of time, very specific neurodevelopmental transitions are happening. And how are we going to figure that out? Well, we're going to figure that out using a multitude of different studies. But I want to remind you of little children with Williams syndrome. People sort of have a whole range of stereotype views of these children. They are hypersocial. They are the opposite of autism. Actually, these are very substantially socially disabled children as well. But there is one thing that happens with these children that is really interesting. They have the Williams syndrome gaze, meaning they are intensely focusing on other people's eyes. So much so that you go here, you go there, and their siblings go here and there, and they focus on the eyes. And they become socially disabled because they're not able to use the gaze cues as a way of learning about the world. They are basically stuck at that reflexive phase of eye fixation. And we are fascinated by this because if that transition is not happening, then it gives us an opportunity to understand mechanistically how a particular assay can clarify pathogenesis in this and in other conditions. So we are hypothesizing things with infant rhesus monkeys. This is like IMAX for monkeys. This is, babies are entirely free, and they are sitting on top of their mothers there, and they are watching. And we are now studying the transitions that happen very early in life when much of the behavior that is reflexive and subcortically guided becomes co-opted into cortically guided behavior. They should leave tracks. They should leave traces of that. So we are conducting both a functional connectivity and tractography study in which we're looking at the transitions, at the formation of those nodes. And just for fun, this is now some MRI data, some diffusion MRI data for that 2-week little guy. And so we're bringing it all together. We're doing those studies with human infants, studying through 3 data points the first 6 months of life of that neurodevelopmental transition in human infants as well as in non-human infants. Let me say a few things about treatment. One of the major challenges that we currently have in our treatments is that we don't have a way of capturing change as a result of those things. That has an enormous impact on some of the things that we do because pharma companies don't want to invest money in big, extended, long clinical trials if they don't have a change of showing very quickly whether or not there is a change. These are my colleagues who are, Larry Scale is the director of clinical trials, and all colleagues who are conducting clinical trials in our center. So very quickly, we had to come up with a way of measuring something, social engagement, in a much more moment-by-moment quantified manner. So if this is the platform for brain development, these are 2-year-olds, then if this is what this is where children learn about the world, we have to be able to measure that. So we modeled the distribution of photoreceptive cells. We borrowed that model from primates, and this is the amount of visual resources that 1-2-year-old is dedicating to that spot on that screen at that moment in time. And the model quickly is that as more and more viewers come together, the colors become hot as there is convergence in where the child is looking, where the children are looking at that moment in time. And so we use that to create really a salience map. And what you're seeing now is basically the same video that you just watch through the eyes of 35 typically developing children. And as you see, it is generating a space-time distribution. As I stop here and there is nothing at all that the babies are paying attention to, and we move on until there is a big convergence. And that convergence means that something really important happened at that moment in time. Okay? So when we sort of quantify this in this manner, and here you have basically one frame of video that are 30 per second, and this is your axis of time, and so you have many frames, and you carve out this maps of salience, what you have is a three-dimensional space-time distribution. And those colors are basically statistics for the amount of convergence that exists among the groups. As we do that, we can then watch the movie and basically quantify what children are engaging with. And what they are engaging with is what their brains are learning about. Remember, brains don't have eyes, they don't have ears. What they have are experiences, okay? And that's what we want to know. We want to know what are the experiences that they're having. One thing that we found that fascinated us is that 2-year-olds, these are typically developing 2-year-olds, spend 80% of the time looking at the same place at the same time. You know, anybody who knows 2-year-olds, I mean, you wouldn't expect that, right? But we call this enculturation. We call this entrainment because this is the basis for shared experiences. Now, here is what we call the funnel of attention, is a significant funnel of attention for typically developing 2-year-olds. And these are now scan paths for those 2-year-olds who have autism, and they diverge significantly. This is the convergence for the typical children. This is the convergence for the children with autism. Watch the same thing, and they're experiencing something entirely different. This is what their brains are changing as a result of. Now, when we took those kinds of many models of convergence, we wanted to know, okay, so this is the climax of that particular video that you watch. And here are the typical children. Here are the children with autism. This is basically what those typically developing 2-year-olds learn from that experience. What did our children learn? Everywhere. So we call this measure a measure of relative entrainment, relative to what is typical. And when we use, what you have here is basically a normative convergence, and those little dots are basically the children with autism. And we use this as a way of quantifying where they are. And we conducted the experiment when the children at 24 months, and it was significantly correlated with their social disability at that age, at the level of 0.45. But look what happened to those children at the age of 14 months. The experiment at 24 months was predicting the way that they would become 1.5 years later at a level of 0.75. Why this was important to us? This was important to us because it was clearly the case that we're not measuring simply the status of the child now. We're measuring the tool that the child uses to learn about the world. And we could actually predict language and cognitive function 1.5 years later, because so much of those skills really, they build on that experience of being with others. Now, we started this work with adults, and I'll show very quickly to you, we have similar methodologies for adults. But if we are to conduct clinical trials that are going to be done quickly, we need to quantify changes in the way that these adults are in the world. We have to quantify that. We have to automate that. And so here is basically the data for adults. We need to find out a significant convergence of attention. This is done mathematically. There is one there. So let's now represent that as a different kind of a curve. So this moves over there, and what you have is that significant convergence translates into that equation. And what you have here is one individual with autism at that particular moment in time. Now, let's compare this with another group of typical adults, and there is nothing standing out. Let's compare this now with a group of adults with autism. Now, the nice thing about this is that this is just one of tens of such significant divergences. So we feel that in this way we can deal with the problem. Now, we feel that we're very close in developing community-viable, objectified, quantified ways of screening children using this kind of developmental social neuroscience. It would be unethical if we could create the best screener in the world, but there was very little that we could offer to these families. So our center has dedicated equal amount of energy and resources to coming up with solutions that are going to provide care for these families where they are. So here are some just brief facts. Less than 20% of children with autism are identified before the age of 3. The Institute of Medicine has suggested reviewing academic center-based sort of treatment programs, that these children require 25 hours of intense treatment delivered by experts. Okay? Now, I don't know about, actually I know the state of New York quite well, but for the state of Georgia, for those 20% that are identified before the age of 3, how many hours those families are given? A week? Not 25 hours, between 1 and 3. Okay? So how can, and you saw, and that's the reason why I showed you that little piece of research a moment ago, you saw that our children are diverging from typical experiences every moment of their lives. So if we want to achieve intensity, if we want to change their path, we have to be able to to provide something that is going to change the experiences of the children all the time. We need to achieve intensity. So remember, if this is the platform for brain development, we got to be able to shape those experiences. So this is what we did in a partnership with Amy Weatherby from FSU, and with the help of Alison Singer is here, who is also helping us with this project. We are focusing on the 3 factors that make a difference in early intervention in the community. And they are the medical home, otherwise called primary care physicians. Every child should have one. Okay? Now, those are the ones on the trenches. They are seeing, all these families for well baby clinics. Every baby needs to come there. Now, if a child was identified in that setting, then it is sent for the early intervention providers, the Part C from IDEA, early intervention providers. This in New York is Babies Can Wait, is Birth to 3. In Georgia is Babies Can Wait. And so we had to find a way of using all of our connections, coming up with a tool that would allow us to train providers where the families are. And here was the idea. The idea was that we're going to use the place where babies and caregivers spend most of the time together in the home as the platform for our intervention. So we had to create a parent-delivered form of intervention in order to become community viable. And so we are now went from tools that train primary care physicians into doing this work and train early intervention providers and support families. And very quickly, because we're running out of time, this is what it looks like. And it provides the training necessary for people to understand what are the signs of autism. It provides them with a platform for understanding the difference between typical development and atypical development. Because believe me, if we're doing that early screening, I don't know if you've seen babies in your life before, if you've done any early identification, but the children with autism that you know these are not the children that we see early on. And one of the big burdens that we have is that we know that the children who are showing less frequent smiling, less this and less that, 5 years later are hijacked by autism. I've seen that. I follow babies over the course of their childhood. We need to change that. So just very quickly, this is the idea.
Autism Navigator is a unique collection of web-based tools integrated into an interactive platform using extensive video footage. This footage illustrates effective evidence-based practice. The Navigator shows side-by-side examples of typical toddler behavior and behavior that may indicate autism.
Armed with visual evidence, parents will feel more comfortable discussing their concerns with their child's doctor.
The Autism Navigator will teach parents intervention techniques that they can use in everyday activities to support learning and development in their child as soon as they suspect their child may have autism.
Fortunately, with early diagnosis and effective early intervention, most children with autism can learn to speak.
So the idea is that we are building on everyday activities that parents and children spend the time doing. And we are engineering an environment in which we are potentiating engagement between parents and children. Is this possible? Gosh, it is. These tools were developed now in environments with single mothers in rural parts of Florida and Georgia. They are now being deployed in Pennsylvania, and they're deployed in Tennessee. And the idea goes from that simple thing that we've learned, that the platform for brain development is early social interaction. But this is a condition that becomes worse with time. In a way, what we call symptoms of autism are symptoms of the disruption of normative processes. What we call the burdens of autism, the intellectual disability, the language disability, the severe behavioral challenges, are things that our children learn because they cannot adjust to the world. By starting early, we are capitalizing on neuroplasticity, we're using what happens in the community to basically support the very platform that is going to make a difference in their lives. And here, the beauty is that, if I was a speech language pathologist, I would be focusing only on this side of the screen. Because now I'm training others to train and to coach. We are now taking care of both sides of this equation. Because if Gordon Ramsay was here and he would give you the first results of what have we learned of the first year of life of these high-risk children, this is what we learned very quickly, is that our children who develop autism, they vocalize just as much as the typically developing babies. What is happening is that those vocalizations are not happening in contingency. So the parents are speaking less and less to their children. It's basically the parents are being trained not to speak to their children because there isn't contingency. So that's a big problem. It's something easily remediated by making people aware and by actually working to engineer that turn taking. This is what we do. Now, I'm very happy with this program because I think that one thing that certainly rejuvenated me in Atlanta is this notion that we've been dealing with autism as an issue of disability for way too long. I think that if we were able to really deploy this, I hope, new system of healthcare delivery, we could change this condition to an issue of diversity. Well, thank you.